Hello everyone, my name is Savanna Vacek, and I'm a sophomore studying civil engineering with a minor in Spanish at the University of Nevada, Las Vegas. My project is on the effect of lanthanides on corn growth. To start, I have my outline, and I will be discussing my background, moving on to the introduction, objectives and hypotheses, methodology, results and discussion, a project summary, and then lastly acknowledgements. To start with my background, phosphorus is an essential element for both animals and plants. It is the battery that stores the energy needed for animals and plants to execute their daily activities. Essentially, without it, we cannot survive. Thus, if water containing phosphorus is released back into the environment in the presence of other elements, it will promote the overgrowth of some species like algae. To overcome this problem, wastewater treatment plants use different methods to remove phosphorus from water before it is released back into the ecosystem. Here you can see in this graphic that if we release the non-treated wastewater back into the environment, due to the excessive richness of nutrients, it can promote the overgrowth of some species like algae and cause the death of some animals from the lack of oxygen. However, if we release treated water back into the environment, we can overcome this problem. To remove phosphate from water, coagulation is a method that can be used. Coagulation is a simple process that involves adding a coagulant that forms large flocks. The flocks are then allowed to settle, leaving clean water to exit. Ferric and alum are prominent coagulants to remove phosphate from a solution. However, there are downsides to using these coagulants. A large amount of sludge is produced when using them, and competition from other usage threatens their price stability. To overcome these issues, we looked for a new coagulant that would be effective at removing phosphate from water. In our search, we found that lanthanides have shown to be more effective than previous coagulants at removing phosphorus from water. In addition, they produce little sludge compared to alum and ferric, which ultimately lowers operation costs. Here in this graphic, you can see the coagulation process. We start by adding a coagulant. Large flocks are then formed. These flocks are then allowed to settle, and clean water is left to exit. The use of lanthanides means animal and plant exposure to elevated amounts of lanthanides. However, the toxicity of lanthanides to plants is barely known. Thus, we propose to study the effect of lanthanides on corn's entire growth cycle. This is important because the study will promote the application of lanthanides in water treatment while promoting water reuse and irrigation. Here in this graphic, you can see that we start our project by growing corn kernels in lanthanide-treated wastewater. Once the corn kernel germinates, we will measure reactive oxygen species, anti-reactive oxygen species, enzymes, hormones, proteins, along with chlorophyll development and the mitotic index. All of these tests will tell us how well our corn plant is doing. Thus, our project objectives are to advance the understanding of lanthanides' effects on corn growth and yield when applied either as fertilizers or during irrigation. We hypothesize that lanthanides can enhance germination and growth chances of corn kernels. We predict this because lanthanides have the same structure and size as calcium, which is known to promote plant growth. Thus, lanthanides can promote kernel germination. We also hypothesize that lanthanides can promote the activity of antioxidant compounds like superoxide dismutase, ascorbate, catalase, and glutathione because of its similarity to calcium again. And also because lanthanides can hold together cell walls and activate enzymes that regulate cellular activities. Moreover, we hypothesize that lanthanides can promote the photosynthetic rate of a corn plant by promoting stomata opening and closure. Although we have different tests to measure different compounds, all of the tests are relatively structured the same. These tests begin by allowing our corn kernels to germinate in the dark for 5 days using different concentrations of lanthanide-treated wastewater. We then crush these germinated kernels to extract reactive oxygen species, anti-reactive oxygen species, proteins, chlorophyll, and enzymes. We then take this mixture and we centrifuge it to separate the supernatant. We then take the supernatant and add additional assays to measure the respective compounds. And lastly, we read the absorbance with the spectrophotometer. In Figure A, root and shoot length as a percentage of untreated seeds was compared with different concentrations of lanthanum. It's evident that growing kernels in a lanthanum solution at less than 100 milligrams per liter actually increase the corn kernel's chances of germination. The root and shoot length increased with respect to the control. At 50 milligrams per liter, the shoot lengths were 16% higher than the control kernels. However, increasing lanthanum concentrations to more than 100 milligrams per liter actually reduced both root and shoot elongation, which is a sign of an unfavorable environment for kernel germination. Roots were more affected by lanthanum concentrations than the shoots. At the highest lanthanum tested conditions, 5,000 milligrams per liter, root length decreased by over 90%, while the shoot decreased by 60%. Root and shoot development is attributed to increased hydrogen peroxide production. Hydrogen peroxide coordinates the beginning of kernel germination, acting as a priming factor that involves specific changes at proteome, transcriptome, and hormonal levels, resulting in acceleration of the germination process. Also, hydrogen peroxide plays a vital role in endosperm weakening and mobilization of seed reserves, which is crucial for seed germination. In Figure B, the excessive production of hydrogen peroxide at 500 milligrams per liter attenuates the germination rate of kernels. Excessive hydrogen peroxide affects membrane structure, function, and enzyme activities, reducing the chances of seed germination. Further reduction of germination at 1,000 milligrams per liter and above is attributed to excessive salt content that damages the cell membranes, lowering the chances of germination. As an overall project summary, we begin with our dewatered wastewater that goes through the coagulation process. Lanthanum is added as a coagulant, and the large flocks are formed. These large flocks are then allowed to settle, and clean water is left to exit. We then take this clean water with different concentrations of lanthanum and use it to grow our corn kernels. After our corn kernels germinate, we study the interaction of reactive oxygen species, anti-reactive oxygen species, enzymes, hormones, proteins, and the mitotic index in the roots. All of these tests tell how well our plant is doing. Moreover, after the plant develops further, we will measure the chlorophyll development in the leaves. Lastly, I would like to acknowledge the UNLV Graduate College Rebel Research and Mentorship Program, along with the Clark County Water District. Thank you.